Neu5Gc sugar is related to the appearance of spontaneous tumors in humans. This is by the Spanish Foundation for Science and Technology Research. Well, what is this Neu5Gc sugar? Where every day we learn something new. A sugar called Neu5Gc, present in red meat, some fish, and dairy products, is related to the appearance of spontaneous tumors in humans. Researchers at University of Nevada, Reno, led by Spaniard David Alvarez Ponce, have analyzed the evolutionary history of the CMAH gene, which allows the synthesis of this sugar, and shown which groups of animals have lost the gene and therefore are more suitable for human consumption and for organ transplants. About 2 million years ago, humans experienced a genetic change that differentiated us from most primates. This change protected us from some diseases, but caused the current consumer products such as red meat to pose a high risk to health. At the time in evolution, the gene called CMAH, which allows the synthesis of sugar called Neu5Gc, was deactivated. This carbohydrate is found in red meat, some fish, and dairy products. If humans consume products derived from animals that have the gene, the body suffers an immune reaction to sugar, which is a foreign substance in the body, and this can cause inflammation, arthritis, and even cancer. Scientists from the University of Nevada, Reno, in the USA, led by Spaniard David Alvarez Ponce, have carried out an analysis of 322 animal genomes to determine whether or not they have active CMAH genes, and next they place the result in the evolutionary tree of the animals to determine at what moment of their evolution the said gene was deactivated. This allowed them to understand why certain species have an active CHMA gene while other similar ones do not. In the first analysis, we scanned all the available genomes. We only found the gene in a few bacteria, in a pair of algae, and in the deuterostomes, a group of animals that include vertebrates and echinoderms, among others. The non-deuterostome animals did not present the gene. And next, we focused on 322 deuterostomy genomes that were available, the Spanish researcher said. The laboratory of Alvarez Ponce specializes in the study of the evolution of genes and genomes through the use of bioinformatics. That is, it's not composed of test tubes, microscopes, or other instruments, but of computers that are used to understand evolution through the analysis of massive amount of data. The toxic sugar present in fish. So far, very few fish species had been studied to know whether or not they had amounts of toxic sugar. Our analysis showed that there are fish that have the CMAH gene and others that do not. But for the moment, the Neu5Gc sugar has been measured in very few of them. In fish that do have this gene, sugar is found in very small proportions in their meat, but in high quantities in caviar. So this sugar is in high quantities in caviar. I bet most of us don't eat caviar that much. Now, this may be because the gene is expressed specifically in eggs or oviducts, says a scientist. Shatisse Perry, the master's course student at the Alvarez Ponce Laboratory, says, it turns out that caviar, one of the most expensive meals in the world, is also one of the products with the highest concentration of Neu5Gc. However, the research also reveals a multitude of fish that do not have the CMAH gene and whose caviar is expected to be free of Neu5Gc. Chicken, turkey, and duck free of CMAH. Like humans, birds also do not have CMAH genes, so consuming chicken, turkey, or duck does not have the negative effects of consuming red meat. Another group of animals that does not have the CMAH genes are reptiles, except for one species of lizard. The presence of the gene in this lizard was unexpected and invalidates the belief, until now accepted, that the gene had been lost in the ancestors of all reptiles and birds, the scientists say. In addition to the above-mentioned food risks, the CMAH gene also plays a key role in the transplantation of organs from animals to humans, a practice known as xenotransplantation, It's one of the factors that determine whether these organs are going to be rejected or not by the human body. When the organ of the animal that has the CMH gene is transplanted to a person, the human body can react to the Neu5Gc sugar and reject the organ, of course. It's possible that the deactivation of the CMH gene during human evolution has protected humans from certain pathogens. For example, there's a type of malaria that needs Neu5Gc sugar to cause infection. This type of malaria affects some primates but not humans, declares Alvarez Ponce. Products to be consumed moderately. The presence or absence of the CMH gene in different animals, which this study has characterized, points out which animals we should not eat, or eat only moderately, and which animals may present pathogenic microbes that affect humans, according to the scientists. If the animal has the gene, then its meat can have the same negative effects as red meat. If the animal does not present the gene, it may contain pathogenic microbes that bind to the Neu5Gc sugar, the precursor to Neu5Gc, and therefore may affect humans. The researchers hope that this study will have a significant impact on later work in the fields of nutrition, genetics, and medicine. To determine in which groups and what moments of the evolution the CMH gene has been deactivated, is critical to know which species are most likely to contain the toxic Neu5Gc sugar, which are recommended for feeding, xenotransplantation, and certain scientific studies, they said. And this is on SciTech Daily by Spanish Foundation for Science and Technology. Please leave your comments and thank you for your support. My Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.